Okay, good afternoon, everyone. I'd just like to welcome and thank all of you for joining the first event in this new virtual series, Food, Nutrition, and Healthy Aging, Lunch with the HNRCA Nutrition Experts. I'm Roger Fielding. I'm the Associate Director of the HNRCA, and I'd just like to share with you a brief description of our mission. The HNRCA is a research institute on the health science campus at Tufts University in downtown Boston. And our scientific mission is focused on understanding the role of nutrition and physical activity on the development and prevention of age-related chronic diseases. We have a very active research community at the HNRCA, and even during this pandemic, we have been working on essential research, and many of us still have been working on our research remotely. I think in no time like the present, we just want to thank you all for your friendship and support of the HNRC and of Tufts, because this really has been an unprecedented period of time during the pandemic. So again, thank you for your friendship and support. I just want to review the format for the webinar. Dr. Ellis will give her presentation in a moment, and then we'll have approximately 15 minutes for questions. We'd like you to submit the questions through the Q&A function on the Zoom link, and you should be able to do that. I'll read the questions, and then Dr. Ellis will try her best to answer them. And again, we'll do that at the conclusion of her presentation. It is really my pleasure to introduce Dr. Jessie Ellis. Dr. Ellis is presently a postdoctoral fellow in the vitamin K laboratory at the HNRCA. She received her PhD in biochemical and molecular nutrition from the Friedman School of Nutrition Science and Policy at Tufts. While there, Dr. Ellis was also a Stanley N. Gershoff Scholar and was also supported by an NIH NIDDK T32 training grant, and also received an award from the Graduate Student Research Award Committee from the American Society for Nutrition at the Experimental Biology Conference in 2017. She also received the HNRCA Cone Student Research Award in 2019. Her research focuses on metabolism of dietary and gut-derived forms of vitamin K and their interaction with the gut microbiome. So without further ado, Dr. Ellis, please.
Thank you, Roger, for that introduction. And thank you to everyone here for calling in and sharing your lunch break with me. I'll get my slides up here. Today, I'm going to be talking about micronutrients and the microbiota and the interactions between those. So this is a topic I'm really excited about. So hopefully today, I'll present something that you guys find interesting as well. So I know we might have a bit of a diverse audience, so I just want to orient ourselves a little bit. When I'm talking about micronutrients today, I'm going to be talking specifically about vitamins and minerals. So micronutrients are critical to human health, and we largely get them from the diet, so they're present in our diet. However, some of these micronutrients are also produced by the microorganisms that live in our gut, or our gut microbiota. So the micronutrients that are produced by the gut consist of many of the B vitamins, as well as several forms of vitamin K that are called menoquinones. Previously our consideration of these micronutrients produced by the gut microbiota has largely been very human-centric, in terms of considering how do these vitamins contribute to our own health. We know that we have micronutrient requirements. We know they're produced locally in the gut. How much do they contribute to that? And while that's an active area of research and a very interesting question, and some of that research is suggesting that they do in fact contribute to our health, particularly the B vitamins. There's a good amount of research that's currently being generated. And some of that contribution might be to known requirements, or it could be some novel health effects going on. But regardless, today I want to challenge you to think about this a little bit differently in terms of a little bit of difference in how these micronutrients might also be used by the gut microbiota, and the micronutrients that are produced specifically in the diet, how that might contribute to the usage in the gut. So, we know that micronutrients are in the diet and these contribute to human health. More and more evidence is suggesting that micronutrients that are produced by the gut microbiota are contributing to human health as well. However, so what I'm going to focus on today is how micronutrients in the diet might also be interacting with the gut microbiota. And that's really what inspired the title of my short talk today, in terms of posing the question, who's feeding who? Because ultimately we're deciding our diet, and so we might be feeding our microbiota nutrients as well. So because it's a bit of a shorter, condensed seminar today, I've organized my slides a little bit with some kind of like bite-sized takeaway points. So some of my slide titles will have that, and I'll summarize them again at the end. So the first takeaway that I kind of want to introduce to you guys is that the gut microbiota have micronutrient needs too. In other words, they're synthesizing them for a reason. And so some of those micronutrients might be used similarly to how we use them. For example, B vitamin or B12 is one of the micronutrients produced by the gut microbiota, and they use B12 as a cofactor in many energy metabolism reactions, which is the same as how we use B12 as well. However, some of these vitamins are used a bit differently. So, for example, vitamin K is another one of those micronutrients that's produced by the gut microbiota. We as humans use vitamin K as a cofactor for vitamin K dependent proteins. The most well-known of those are the proteins that are involved in blood coagulation. However, micronutrients use them differently. So the microbiota use vitamin K actually as an electron carrier in energy production, anaerobic respiration. So essentially energy production in the absence of oxygen. So it's a very different usage. But even though the microbiota can synthesize some of these micronutrients, they can't always synthesize all the micronutrients that they need. So this is a figure from a paper that showed basically all of the strains of bacteria within the human gut microbiome, and all of these colors represent encoding the basically metabolic pathways for synthesis of each of these vitamins. So B1, 2, 3, 4, 5, sorry, not 4, 3, 5, 6. And basically what this is showing is that you can see by the white gaps here, not all bacteria can produce all of these micronutrients. And sometimes they require nutrients that they can't necessarily produce. So the next, and so the second takeaway that I want to present here is that the gut microbiota compete for and share micronutrients. So this is a bit of a simplified diagram, but I like how it illustrates it. These are some nutritional interactions between microbiota or bacteria for certain nutrients. So for example, two different bacteria might compete for the same nutrient. This is a relationship called syntrophy, in which the production or the metabolism of one micronutrient, there might be a byproduct that is then utilized by a second, or there could even be cross-feeding, where the product of one bacteria is fed to the other. And we can think about this in terms of competition or sharing of micronutrients, but also keep in mind that these are interactions that would shape also the bacteria and the relationships between them. For the cross-feeding, it can also, there are different levels of complexity. You can also have something called mutual cross-feeding, where two bacteria have a relationship where they feed each other different products. And you can think of this as a very mutual relationship where they're encouraging each other's growth. And in terms of using an example with a micronutrient, within the human gut microbiome, many of the Bacteroidetes species encode biosynthesis pathways for all the B vitamins except B12. So thinking back to that circular diagram, they would encode for all of them except B12, whereas the firmicutes often include just B12. And so you can imagine that these bacteria are actually swapping vitamins and feeding each other and supporting each other's growth. And so to use, again, a more specific example, there's been a lot of really interesting research done with B12. So B12 is a coronoid. So a coronoid are B12-like molecules, and gut microbes can synthesize a variety of these different coronoids. They use them similarly in cofactor roles, but they're structurally a little bit different. So B12, so this red here, and then coronoids are others. You can see that bacteria actively synthesize them, actively swap them, and then this competition also might shape the gut microbial communities. And so this is showing the same in a little bit of a different way, community of bacteria. But what I really want to emphasize here is these researchers, this was a publication in Science just earlier this year. If you look at the B column here, this is the human gut bacterial genomes. And what they found was that of those 311 bacterial genomes, about, you could say about 25 or 30% of bacteria can both produce cobamines and use them. About 50% use them, but they don't produce them. So you can, so they're obviously getting them from somewhere else or have dependencies from either, gut microbes or the environment. And then maybe about 20% don't use them at all. So the reason I wanted to present this is because this is a significant amount, more than 80%, it looks like, of the human gut bacterial genomes that were characterized here have a dependency on cobamides, which just, I guess, is just driving home the point a little bit that bacteria do have these dependencies on micronutrients. So the third takeaway here is that in consideration of this, I've talked about these interactions between the microbes, but what I was focusing my talk on today is how dietary micronutrients also influence the gut microbiota. So the gut microbiota, both composition and function, have been shown to be disrupted by deficiency. So vitamin A, iron, zinc, folic acid, in all of these cases where these micronutrients are absent in the diet, the composition of the gut microbiome is altered. And I want to point out here, some of these micronutrients are produced by the gut microbiota, such as folic acid, that's B9, that's one that some species in the gut can make. But vitamin A, iron, and zinc are not microbially produced nutrients, yet the bacteria in our gut are dependent on them. So again, it's just, I guess, drawing this relationship between the diet and the gut microbiome. So this is a bit of an interesting point too. So a couple of years ago, a group showed that some bacteroides in the gut have what they termed acquisition machinery for B12. So in humans, we have intrinsic factor, which binds to B12 and helps us absorb it. Some bacteria in the gut have machinery that can remove intrinsic factor and basically strip it off and to make B12 available to themselves, which is actually pretty interesting. And so this sparked some questions about, are we competing with our gut microbes for micronutrients? And where it's possible, I guess the counter to that some people have raised is that largely a lot of the absorption of dietary micronutrients for us occurs in the small intestine, so further up in the intestinal tract, whereas the majority of our gut microbes live lower down in our gut. So, basically they could be getting our scraps or what we don't absorb, but there is the possibility for some competition, especially in conditions where there's infections or where bacteria are colonizing maybe further up in the intestinal tract than they should be, and there could be some competition there. But just a bit of an interesting side there. But in terms of how these bacteria are getting micronutrients from either each other or the diet. So bacteria have transport mechanisms for vitamin acquisition, for vitamins that they don't produce themselves. So you can imagine if they're not producing it themselves, they're going to have to acquire it and transfer it through their bacterial membrane. So to the right, I have just a couple examples here. So thiamine, this is vitamin B1. OM here stands for outer membrane, inner membrane on the bacterial wall. And there are several characterized transporters to import thiamine from the environment. And so about half of gut microbes are able to synthesize vitamin B1 themselves. but many of them have instead these transporters to either acquire thiamine or a precursor, and then they can finish the synthesis themselves. Similarly, for B12, about half of gut microbes have these transport mechanisms. And so, it raises, the question, or one thing we can think about is, if these are micronutrients that are needed by bacteria, and, why would they trade off making them themselves for getting them, for a dependency either on other bacteria or the diet? And one possible reason is just that making these vitamins themselves can be energetically very costly. So you can see here, and you can appreciate from this, B12 is a quite complex molecule. There's about 30 enzymatic steps to make it from scratch. And so some bacteria might opt instead to transport it in from the environment, whether that be from another bacterium or from the diet. So the next takeaway that I want to share is that the gut microbiota can actually remodel some micronutrients. So I mentioned before that B12 is a vitamin in the coronoid family, and that bacteria, you synthesize these molecules. There's slight variations on it, but they all serve a similar purpose, but some bacteria prefer one form or another. So this again is the structure of B12 here, but this group here can be swapped out for a number of different groups. And so some bacteria might prefer this, whereas others might prefer this. And what's really interesting is that it seems that whether bacteria are getting this either from the diet or from each other, they're able to remodel it. In other words, remove this and replace it with another group of their choice for their own metabolism. And according to some estimates, about up to 80% of the B12 that is not absorbed from the diet might be remodeled to these other coronoids. So then the last takeaway I want to share here is a little bit of a broader consideration, but also a bit of a summary. It's not just the good guys. So pathogenic or bad bacteria utilize micronutrients as well. So I want to show this diagram here because it summarizes a couple of the points that I've talked about here. So it shows here, this is the, you can imagine this is the inside of the gastrointestinal tract, where this is the lumen, or kind of that open area. These are our intestinal cells. Dietary and supplemental vitamins might be used by commensals. So I want to, just make sure I define this too. Commensals are the bacteria that are, the good guys. They're beneficial to our health. So there are a couple of relationships that we see here. So I talked about some of those transport mechanisms. This is one of those transporters for thiamine. You can imagine that some of these bacteria might be transporting in vitamins either from the diet. You can also see here that there's an example of some cross-feeding. So this bacteria might be making B12. This bacteria might be taking it up, again, by another transporter. And then you even have some of these more complex relationships here where they're trading. So this bacteria might be making thiamine, this one might be making folate, and they're supporting each other's growth. And then also, there's the contribution to human health where either from the diet or, potentially from some of the bacteria as well, we're absorbing those vitamins as well. So this would be in a healthy state. However, these types of relationships can also go wrong, so to speak, if the wrong bacteria are there. So if we think about if the presence of a pathogen or a bad guy, they can use these vitamins too. So they could outcompete. So if they require the same vitamins, they might outcompete the good guys and disrupt these relationships that then also have impact on the host. And so I wanted to summarize this just to, point this out. This is a really interesting topic to me, but it all depends on the environment as well that's currently going on. And just if we're thinking about supplementation or anything like that, you have to think about the existing state as well. So in summary, I just wanted to put these key takeaways up again. The gut microbiota, they have micronutrients too. They share for and they compete for and share micronutrients. And they also, seem utilize dietary micronutrients, and they can remodel some of those as well. And this happens with both bacteria that are beneficial for us as well as pathogenic bacteria. So with that, I want to thank you guys for joining, and I'm happy to take any questions. I saw some of them rolling in as I was speaking, so happy to take any questions and hope I can answer them.
Thank you, Dr. Ellis, for a really excellent presentation. For people who are interested in asking questions, please submit them through the Q&A function. And I will read the questions, and then we'll see if we can answer them. A few questions about the slides, but those are all up now. I guess let me start off, Jessie, while people are submitting their questions. You know, is there ways to change the sort of makeup of your gut microbiota by taking probiotics or taking different dietary patterns or supplements? Do you know?
You know, it's interesting because that's it's a really it's an important question. You know, they have seen that different you know, makeups are associated with different dietary patterns. That's one. So, you know, diet is an important shaper of that, but it might be harder to change than we think. And also, another important consideration is just some of that foundations of our gut microbiome are formed early on in life. So the composition might be more malleable early on and harder to change later. But diet definitely does affect it. Whether or not it's a strong enough lever to really change it in significant ways without other factors, it's hard to say. You know, I think in terms of at least of a community, you can think about other dietary interventions such as, you know, increasing your fiber. You know, that increases, you know, several, you know, beneficial species. So there are some triggers or, you know, some things I think that can change it. I think overall, community shifts are a little harder, but there are definitely some levers we can pull. But also, you know, there are so many other lifestyle factors that affect the composition as well. That is kind of sometimes hard to say what's the biggest driver.
Well, let's go. There's some more questions popping in, so let's get to some of these. This question comes from Linda Holt. Does the cycle change as we age? Are there age-related changes?
In the gut microbiome composition, definitely. We do know that it changes as we age. One, I guess, of the biggest factors is that diversity decreases as we age. So typically, you know, speaking very generally, more diversity the more diverse the gut microbiome, the better. We do see some change or some decreases in diversity as we age. So.
Okay, Wilbert Sebesma asks, would you favor colon-targeted delivery of vitamins for certain states of microbiome dysbiosis? So I guess trying to surpass maybe.
Yeah, you know, that's an interesting question as well. I think, you know, it's kind of going back to one of those last slides. It's challenging because it depends on the dysbiosis, you know, right? So whatever that imbalance is, sure, you could target certain micronutrients to the gut, but I guess it would require a lot of knowledge about this, one, the species that are already there, and two, their metabolism. So if you're supplementing a vitamin or a mineral or something, you want to make sure that it's benefiting the good guys more than the bad guys, which is, I think, really interesting and a direction I can see this field going in terms of just personalized nutrition. But that requires personalized background and knowing what it is to really be able to target that. But another thing I would say is, you know, you could, you know, design a supplement or something like that to you know, not be absorbed or something, you know, kind of like they do with drug delivery, where you'd want to target it to the gut. But another thing I would say is that with a lot of micronutrients, you know, they have transport mechanisms where they can become saturated. So if you give a larger dose, a decent amount of it's probably making it down to the colon anyways. So I'm not sure you need to design something specific.
Great. So Valerie asks, whether we're feeding our cells or our microbes, how does that impact human health? So are there examples of how this may affect health?
Yeah, I mean, so I didn't talk as much today about the other half of that being the impact on human health. And part of the reason I didn't is because that's a hard question to answer. You know, in the terms of micronutrients and how much we're absorbing, that was kind of one of the questions I showed a little bit with. Some of those micronutrients that are produced by gut bacteria, we might have transporters then in our colon to absorb them, and those would be direct contributions, right? So contributing directly to our need for B1, or, you know, for thiamine or folic acid or something like that. But there are also so many indirect effects because the gut microbes are doing so much more than just vitamin production. You know, they're producing other metabolites as well as some of those relationships where it's, you know, they might be producing other metabolites that are either, you know, helping maintain the integrity of our gut to make it more resistant to pathogenic bacteria, or either other health effects we don't even know. So I guess it's a little bit hard to answer specifically in terms of you know, micronutrients and how it's contributing to health, because I think that's why the field is just kind of exploded right now, is because there are so many ways, potential ways that they could be impacting our health.
Yes, Katrina Sarson says the talk is very interesting and she wants to thank you, but she asks, have you observed any change in microbiota activity through the aging process? And do you have any specific dietary recommendation based on your finding? I guess you answered the age question, but what about the dietary recommendations? Are there
Yeah.
diets
So
with good microbiota?
yeah. So I guess I would say that's in terms of, you know, specific recommendations. That I don't work so much on that, but I guess what I would say is, you know, there are several things that that very generally, you know, can support a healthy gut microbiota. And I think, you know, it kind of goes sometimes back to the same advice we always hear: fruits and vegetables, you know, fiber, some of those micronutrients, they provide a lot of substrates that the good bacteria need. So yes, even though diversity declines, you know, I think just, you know, speaking very generally, a healthy diet supports a healthy gut microbiome.
Paul asks, how much do antibiotics used for treating illness affect gut microbiota? And if so, how can this trade-off be managed?
Yeah, that's a great question, because, you know, you know, Roger asked about, you know, how much does diet influence the gut microbiome, and it does. Antibiotics are one of those things that really are influential on the gut microbiome. And depending on, you know, which different antibiotics target different bacteria, you know, how best to mitigate that. I guess, you know, one of the things we can do, and I know this is an effort in the medical field in general, is not taking antibiotics for things that aren't necessary. But of course, in the cases where it is, you know, that's I think treating an infection in which an antibiotic is necessary is the primary concern. But in terms of rebounding from that as well, I do know that's one instance where probiotics can be really beneficial in kind of helping seed some of those microbes back in a healthy state. You know, when you already have a very, you know, established community, probiotics, or, you know, introducing other bacteria can be, you know, minimally effective sometimes, because the bacteria that are already there out competing what you're adding. But in a case where you've wiped out a lot of those with an antibiotic, supplementing a probiotic can be more beneficial, just because, you know, they actually have more of a chance to take root.
So here's a question I think a lot of people have, Jessie. Allie Gallup asks, do you see any benefit to consumers using test kits to evaluate what bacteria / community of bacteria they have? Any research to show that that's helpful to use when you get this info creating individualized nutrition programs?
Yeah, so I am trying to think of the guy's name. There is some really interesting work. So I guess what I would say is it does provide some information, whether or not it's, you know, necessary for everyone to get their microbiome assessed, and no, probably not. But it is pretty interesting, I guess, if you're trying to tweak certain things. So for example, I'm thinking of, and I'm blanking on the guy's name, but he does some research with personalized nutrition and the gut microbiome. And for example, showing that characterizing the gut microbiome can help better predict things like glycemic responses to specific foods and things like that. So if you are trying to optimize your nutrition or things like that in terms of, what foods are really spiking my blood sugar, or things like that, that might actually be involving the gut microbiota, something like that can be really interesting. And just being able to monitor, for example, your blood glucose response in response to certain foods being predicted by your gut microbiome. That could be a really interesting thing. There are some examples like that. So I guess for the consumer who's really interested in targeting their nutrition and diet like that, definitely. But is it necessary? No, but in the future, when it becomes more widely available and easy to do and everyone can do it, sure, I think that'd be really cool.
Thank you. So your former colleague, Ken Westerman, congratulates you on the presentation and
Thanks,
asks,
Kenny.
how stable in practice is the good bad guy distinction? The other angle will be effective bacteria in highly individualized way depending on the specific ecology of each person's gut microbiome.
Yeah, and it's sometimes harder than we think. There are so many organisms that live, in our gut, and to categorize them as good or bad is probably too simple of a categorization. And again, I think it just kind of depends on, the rest of the gut environment. Obviously, there are some bacteria, that like cause infections, or take over. But in terms of, do I think we can point to every single microbe that we can identify in the gut microbiome and say, he's a good guy, he's a bad guy? No, definitely not. But I think that eventually we will be able to, at least, in terms of different situations.
Okay, Sai Das asks, and also compliments you on your talk. That's a recurring theme. Do you think or know if some of the increased neurodegenerative diseases are potentially attributable to changes in diet and hence the balance of microbiota requiring B12 in particular and other important B vitamins needed by humans?
That's an interesting question as well, and I guess one that I don't think I can answer, but I can, I guess I can speculate on as much as we can. But that's a possibility, and especially for some of these B vitamins, we have seen that it does appear like we can absorb some of them from the gut, and so yeah, maybe some of these vitamins are involved or contributing to our own requirements or, prevention of certain, neurodegenerative diseases, you said. Another thing that question made me think about is some of our like neurotransmitters, and I've seen some estimates on like the amount of serotonin and things that are even produced by the gut. And it's quite large. So it's an interesting question, and I'm not sure we have that answer yet, but in terms of could they be contributing, I think it's possible, yeah.
Okay, there's a few questions that are all relatively similar, and I think they sort of ask the question about, can you actually diagnose sort of gut microbiota dysbiosis now? And I think some of them ask sort of in a different way, but are there ways to classify what's good and what's bad in your gut microbiota? I mean, where is the sort of state of the art with that now?
Yeah, so there are some, it's challenging because kind of going back to that other question of just like, can you say can you absolutely categorize whether some are good and some are bad? Some of what also might depend are the relationships to each other. So I feel like even thinking back to some of those earliest kind of like characterizations of like Bacteroides to firmicutes and how that relates to energy utilization and how much energy is actually liberated from in conjunction with the gut microbiome as well, and associations with obesity and things like that. I don't know that at this point, if we took a sample, we analyzed it, and we can say, for certain that this is very good and this is bad. I think it kind of depends like what you're looking for. And I'm trying to think of, I guess, another way to say this, I think that in certain, instances, if a person is, presenting with symptoms, gastrointestinal symptoms, and you can then, in that case, you could look at it and say, yes, like you have an overabundance of this bacteria that we know to be associated with gastrointestinal discomfort, or things like that. So I think definitely in terms of some of the more common, either infections or dysbiosis, even some things with like IBS and things, there are certain strains that have been identified to be, contributing to that. But in healthy individual, I think that's hard. But definitely, we also know that there are some strains that we've, categorized that we know that are good. You think about, like, especially, like bifidobacteria and things like that, that we know to be good, and lactic acid bacteria. But I think that there are also so many different combinations of those good guys too.
So Tammy Silver asks, what are the lifestyle factors that affect microbiome composition?
Everything. I think that's what's hard. There's diet, exercise, stress, sleep, just about everything impacts your gut microbiome, but which, I think kind of goes back to, well, it's maybe a bit of an oversimplification, but if you just think about if it's good for you, it's probably good for your gut, because you're the one, you are creating that environment, for your gut. So if the environment is healthy, the microbes are going to be healthier too. In a very oversimplified explanation for that.
Okay, Barbara Eisenhower asks, many probiotic supplements are single strain, usually Lactobacillus acidophilus, for example. Mm-hmm. After taking an antibiotic, would a single strain supplement be okay to take, or would a variety of strains be better?
Again, I think it kind of would depend on the specific antibiotic, because different antibiotics are wiping out different bacteria. They're targeted for different bacteria. So, sometimes you might, even if you're receiving antibiotic treatment, be receiving like an antibiotic cocktail. I'm not a clinician, so I'm not sure I can, answer that with a lot of specificity either, without knowing the specific antibiotic. But my intuition would be that probably a more diverse probiotic would be more advantageous, just because you'd want to colonize, more of a community rather than a single strain. On the other hand, if an antibiotic is disproportionately knocking something out, maybe you'd want to supplement with that. But again, I would kind of refer to a clinician on that.
Good answer. Marianne Anderson asked, this is an interesting question. I think if you might want to, if you don't feel comfortable answering it, so I don't think you should. But how does the bad guy H. pylori affect B12 in the gut?
Oh, so H. pylori, it's Helicobacter pylori, causes a lot of, so it's more associated actually, I think, with like gastric, more in like the stomach, causes inflammation in like the stomach lining, more so than the lower intestine. So in terms of B vitamins, I do think that, so I'm actually thinking about a paper that I was on, we worked on with a group at MIT, that they were looking at supplemental folate actually in prevention of H. pylori. So I guess what I'm saying is I'm not sure exactly if your question is asking more about competition for it, or if supplementation could prevent it, because I know that there is some research, at least basic, like in animals, that was exploring supplementation of folate as protective against H. pylori. So I'm not sure if that answers your question or not.
And then I think maybe, because it's sort of coming up on time, maybe this is one final question, Jessie, and I think it's interesting. Valerie asks, what about gut microbiota dysbiosis and obesity and possible treatments?
Yeah, I think that, again, that's interesting, because the relationship is so, reciprocal, that, we're affecting the gut bacteria and they're affecting us. So sometimes it's hard to say what the cause of dysbiosis is. And that makes it challenging to treat too. So I definitely think, though, that some of that if there are some contributions, from the gut microbiome, so I guess complications of obesity that are contributing to dysbiosis, if you could somehow design strategies to treat that, cyclically, that could help as well and improve health. But I don't know any specific strategies to mention.
Okay, well, thanks for some great answers to those questions, Jessie. I'd just like to sort of close our session by thanking you for an excellent presentation and really thanking all our friends and supporters of the HNRCA and Tufts for attending and participating in this inaugural Healthy Aging and Nutrition webinar. We are very excited about the fact that the series will continue in the new year, so please keep an eye out for updates on future scheduled events in this format. And as I said before, we always thank you for your continued support for the HNRCA and Tufts. And if you are interested in supporting us, there is a link to Tufts Giving in the chat feature to support the work of the HNRCA. So without further ado, again, I want to thank all of you for joining us today and for the wonderful presentation by Dr. Jessica Ellis. Thank you all and have a great afternoon.
Yay.
Okay.